Hello students, welcome back to our video sessions on class 9 biology. We are into our third chapter now, which is about animal tissues. In the previous lesson, we learnt about plant tissues. Now we are going to learn about animal tissues. There are more than 100 visibly distinguishable kinds of differentiated cells in the vertebrate animal. These cells are organized into tissues and the tissue is organized into organs. Groups of organs make up various systems. There are four types of tissues in animals. They are the epithelial tissue, connective tissue, muscle tissue, and the nervous tissue. Let us first study about the epithelial tissue. The epithelial tissue lines body surfaces and cavities. They cover internal organs. They also form glands. The cells of the tissue are closely connected to each other. The epithelium is found on edges of organs. So, it has an apical surface which is exposed to the body cavity or exterior and a basal surface adjacent to the underlying tissue. Epithelial cells can be one of the three types. They may be squamous, flattened, cuboidal, or columnar epithelial cells. If the epithelium consists of a single layer, it is called simple epithelium. If it has more than one layer, it is called stratified epithelium. So, depending upon the cell shape and the number of cell layers, epithelial cells are classified. Example, simple columnar epithelial tissue will have one layer of column-shaped cells. Then you have the simple squamous epithelium in the inner lining of blood vessels. They decrease friction. They are also present in the walls of alveoli in lungs. The next type is simple cuboidal epithelial tissue. It is there in the tubules of the kidney. It helps in absorption and secretion from or into the kidney filtrate. The next type is the simple columnar epithelial tissue. It is there in the intestinal lining. This tissue helps in absorption of nutrients. The last type is the stratified squamous epithelium, which is there in the mouth, esophagus, and the skin. This epithelium helps in protection against abrasion. The next type of tissue is the connective tissue. Connective tissue is the tissue that connects and supports all other types of tissues in the body. Like all other tissue types, it consists of cells surrounded by extracellular matrix. It is different from other tissues in that its cells are loosely packed within the extracellular matrix. The three components of the connective tissue are the cells, ground substance, and fibers. Ground substance and fibers make up the extracellular matrix. The primary cell of the connective tissue is fibroblast. Its function is to maintain the extracellular matrix of the connective tissues. Other cells are cells of the immune system and adipocytes. Then there are specialized cells like in cartilage there are chondrocytes and osteocytes in bone. So there are cells that make up the connective tissue. Then the ground substance which forms the extracellular matrix is a viscous gel made of water, proteoglycans, glycoproteins, and glycosaminoglycans. They bind high amounts of water. They help in nourishing the tissue. And then there are protein fibers which are collagen, elastin, and reticular fibers. So the ground substance and the fibers make up the extracellular matrix. Now let us learn about various connective tissues. We will be learning about the areolar tissue. Then we will be learning about the bone, cartilage, tendon and ligament. And finally, blood is also one type of connective tissue. So now what is the areolar tissue? It is a loose connective tissue which contains equal amounts of cells, fibers and ground substance. However, immune cells are also present. This presence of immune cells helps this tissue to fight against pathogens. The principal fibers present here are collagen fibers, which are sparsely distributed within the extracellular matrix. That is why we call this as a loose connective tissue. Reticular and elastic fibers are also there. The areolar tissue is the most widely distributed type of connective tissue lining the body's inner surfaces. It is flexible but not very resistant to mechanical stress. It binds tissues to organs, holding organs in place. It attaches epithelial tissue to other underlying tissues. Cells of the areolar tissue are fibroblasts, which are widely distributed in this tissue. Example of the loose connective tissue is the lamina propria of alimentary canal and respiratory tracts, mucous membrane of reproductive and urinary tracts. The next type of tissue we will be learning is the adipose tissue. It is a specialized connective tissue. It consists of lipid-rich cells called adipocytes. The main function of adipose tissue is to store energy in the form of lipids. This tissue is present under the skin, which is called parietal, or it is present surrounding organs, which is called visceral. There are two types of adipose tissue. One is the white adipose tissue, which is seen in adults, and then there is a brown adipose tissue seen in newborns. The other functions of adipose tissue is to cushion the organs, and it also helps in insulation. Next, bone is a connective tissue containing cells, fibers and ground substances. There are three types of cells in the bone connective tissue: osteoblasts, osteocytes and osteoclasts. The bone forms the framework that supports the body. Bone is the major component of the skeletal system of several vertebrae. It is made up of organic matrix and inorganic matrix. The inorganic matrix consists of calcium phosphate and calcium carbonate. These salts give hardness to the bone. They are secreted by osteocytes. They are present in the lacunae, that is the spaces within the bone matrix, and they help in maintenance of the bone. The next type of connective tissue is the cartilage. Cartilage is a flexible connective tissue found in joints of bones, tips of the ribs, tip of the nose, external ear, and in the trachea. The primary cell type is the chondrocyte. There are three types of cartilage. One is the elastic cartilage, hyaline cartilage, and fibrocartilage. In 8th class, you must have studied about the thyroid cartilage, which forms the Adam's apple. So, the thyroid cartilage is one type of hyaline cartilage. The next connective tissue is the ligament. Ligaments are a band of dense regular connective tissue bundles made up of collagenous fibers. Ligaments connect bones to other bones to form joints. Tendon is a type of connective tissue which is also made up of fibers. The tendons join muscle to the bones, and it is also made up of collagen. Blood is a fluid connective tissue. It is a tissue and a fluid. It is a tissue because it is a collection of specialized cells that serve particular functions. These cells are suspended in a liquid matrix called plasma. Thus, blood is a fluid too. Coming to the composition of blood, blood consists of plasma and formed elements. The formed elements are the cells. The plasma has organic substances and inorganic substances. The organic substances are the amino acids, proteins, and nutrients. The formed elements of blood are the RBC, which is also called erythrocytes, WBC, which is also called leukocytes, and then there are platelets, also called thrombocytes. The leukocytes are again of two types. There are granulocytes and agranulocytes. There are two types of agranulocytes, the lymphocytes and monocytes. The granulocytes are neutrophils, basophils, and eosinophils. The red blood cells make up 45% of the whole blood. The plasma makes up 54.3% of the whole blood, and the WBCs constitute 0.7% of the whole blood. The RBCs or the erythrocytes are most numerous out of all the formed elements. They are tiny biconcave disc-shaped cells. The primary function is to transport oxygen. There are 4 to 6 million cells per cubic millimeter of blood. The blood is red in color because of the presence of a pigment called hemoglobin. Each RBC lives for 120 days, after which they are destroyed in the liver and spleen. So the spleen is also called the graveyard of RBC. Mature RBCs do not have a nucleus. Then talking about the WBCs, they are also called leukocytes. They are larger than the erythrocytes, but they are few in number. They have a nucleus, but they do not have hemoglobin. These help in immune response. The neutrophils help in phagocytosis of foreign bodies. Macrophages swallow and destroy cell debris and bacteria or viruses. The lymphocytes, there are T cells and B cells of the lymphocytes, they fight infection. The monocytes attack and engulf foreign substances. So these are the leukocytes. Now next one is the platelets. Platelets are also called thrombocytes. They help in blood clotting. When there is any injury, the platelets accumulate at the site of injury and help in formation of blood clot. The clot seals the wound and prevents further blood loss. The human adult has approximately 5 liters of blood, and this fluid is pumped by the heart through various blood vessels called the arteries, veins, and capillaries. The heart pumps nearly 36,000 liters of blood in the distance of nearly 20,000 kilometers in the time period of 24 hours. This information is just to get an idea of the volume of blood and the efficiency of the heart. Blood groups were discovered by Landsteiner. There are 4 main blood groups as defined by the ABO system. The various blood groups are blood group A, blood group B, blood group O, and blood group AB. The blood group A has antigen A on the surface of the RBC, and it has anti-B antibody in the plasma. Blood group B has antigen B on the surface of the RBC and anti-A antibody in the plasma. Blood group O has no antigens, but it has anti-A and anti-B antibodies in the plasma. Blood group AB has antigens A and B, but it does not have antibodies. Compatibility can be defined as the property of one blood group that allows it to come into contact with another blood group without inducing adverse reaction. The adverse reaction is an antigen-antibody reaction. To understand this, let us take an example. Group A recipients have A antigen on their red cells. So they cannot receive group O or group B, as anti-A will attack their red cells. Group B recipients have B antigen on their red cells. So they can't receive group O or group A plasma, as anti-B will attack their red cells. Group AB recipients can only receive group AB plasma. Group O recipients do not have antigen A or antigen B, so they can safely receive plasma of any blood group type. So, because AB group human beings can receive blood from any other groups, they are called universal recipients. O blood group people can donate blood to any other group, so these people are known as universal donors. How do we identify our blood group? This is a small experiment which requires a blood identification kit, which is also called blood grouping kit. We need a glass slide, a wax pencil, disposable needle, cotton, toothpicks, and 70% alcohol. So the blood group kit will be having anti-A sera, anti-B sera, anti-D sera, a porcelain white plate, there will be a wax pencil, a needle, and you will also have a manual. So for the procedure, we will take a porcelain plate or a tile and clean it dry. The plate must be clean. Using a wax pencil, we will draw 3 circles on the plate to divide the surface into 3 different parts. We will place 1 drop of the corresponding anti-serum near the edge of each circle, as we can observe. After preparing the glass plate, we will choose a finger, usually the left ring finger is chosen. This fingertip is cleaned with alcohol using a cotton ball and then it is let dry. The finger is pressed to bring the blood to the tip of the finger, and quickly the fingertip is pricked with the help of a needle. The needle should be a sterile needle. 1 drop of blood should get into each of the circle without touching the anti-sera. After putting 3 drops in each circle, the finger is again pressed with cotton ball so that excess blood will not leak out. And using a toothpick, the blood is mixed with the anti-serum gently. So in each circle, the mixing is done with a different toothpick. The toothpick should not be reused after mixing it in one circle. We should watch clearly in the circles for agglutination or clumping. Agglutination will appear as grainy clumps of red blood cells suspended in a clear solution. So, observing the agglutination, you can identify the blood group. If agglutination occurs in anti-D serum, then the blood group is Rh positive, and if it does not, then it is Rh negative. If there is clumping in anti-A and there is no clumping in anti-B, then the blood group is A type. If there is no clumping in anti-A and clumping in anti-B, then the blood group is B blood group. If there is clumping in both anti-A and anti-B circles, then the blood group is AB blood group. If there is no clumping in both the circles, then it is blood group O type.